Well, hello and welcome to our Hearing Health Foundation research webinar. I'm coming to you live from New York City, where it's a beautiful, crisp, sunny fall day. I hope that you're also having a very sunny afternoon, wherever you may be. Now, please do enable the Zoom captions by clicking the CC button in the toolbar at the bottom of your screen. These captions can be moved anywhere on your screen, and if you need any other assistance using Zoom, please follow the link to the technical guide shared in the chat. Also, please note that this event's being recorded and will be posted with captions. Now, by way of introduction, my name is Dr. Anil Lawani. I'm a professor and vice chairman for research in the Department of Otolaryngology, Head, Neck Surgery, or Ear, Nose, and Throat, as well as Associate Dean for Student Research at Columbia University, Vagelos College of Physicians and Surgeons in New York. I'm also a board member at Hearing Health Foundation and the head of HHF's Council of Scientific Trustees. The council oversees the Emerging Research Grants Program, affectionately known as ERG. Now, ERG is a competitive program that awards funds to researchers conducting cutting-edge hearing and balance research. These grants supported many leaders in our field to become successful scientists, including Dr. Vélez-Ortega, our speaker today. Now let's get to the topic of our webinar today. So the cochlear epithelium has two types of auditory sensory cells known as the inner and outer hair cells. They get all the glory, but they're surrounded by nearly a dozen different types of supporting cells, all organized in a very particular pattern. Now, the hair cells are responsible for converting sound vibrations into electrical signals, and thus most cochlear auditory research is focused on these sensory cells. In contrast, much less is known about the different types of cochlear supporting cells, even though they're likely to impact hair cell function. In this webinar, we're going to take care of this weakness by having Dr. Catalina Vélez-Ortega describe how, after noise exposure, several types of cochlear supporting cells are involved in detecting tissue damage and in regulating hearing sensitivity. Now, Dr. Vélez-Ortega is an assistant professor of physiology and otolaryngology and head neck surgery at the University of Kentucky. She was a 2018 Emerging Research Grant Scientist generously funded by Cochlear Americas. Now, Hearing Health Foundation's ERG program that provides seed money to scientists who are starting out in the field of research is only possible through the generosity of supporters like you. Now, if you'd like to support our work on hearing loss, tinnitus, and related conditions, you can do so today at hhf.org/donate, hhf.org/donate. And now we'll move to Dr. Vélez-Ortega's presentation. And please do ask your questions through the Q&A box linked at the bottom of the screen, and we'll try to answer the questions following the presentation. Dr. Vélez-Ortega.
Well, thank you for that introduction, and thank you for the invitation to share my work at this webinar. So, like we just heard, we'll be looking at different types of cells in the cochlear epithelium. Here's a picture from the mouse cochlear epithelium. The cochlea is this nail-like structure, this spiral organ that converts sounds into electrical signals. So here I'm just showing half of one of those turns in the cochlea. And in white or gray are the nuclei of all the cells in the tissue. And in cyan, we have a particular label for the hair cells, the stars of the tissue, the ones that detect sound. If we zoom in, we see that these hair cells are highly organized in rows. A cross-section through this tissue, so like a side view, will show again one row of the cells, a space, 3 rows of the other cell types. Now here below I have a cartoon showing what these cells are. The first row, the inner hair cells, and then this space, which is a fluid-filled chamber called the tunnel of Corti, and then 3 rows of outer hair cells. And here in these pictures, again, the nuclei of all these surrounding cells are shown in white or gray. All of these other cells are collectively known as supporting cells, cochlear supporting cells. But they're actually different types. They have been characterized based on their morphology, localization, they express different genes, and they're very organized around the hair cells. For instance, this fluid-filled compartment, this triangle here, the tunnel of Corti, it's made up of the inner and outer pillar cells. Now the hair cells are sitting on top of, the outer hair cells are sitting on top of Dieter cells, and we have here Hensen cells. So there's multiple types of supporting cells, but very little is known about them. Most research is focusing on these hair cells. In fact, half of my lab is studying these organelles on top of the hair cells that detect sound. The other half is studying these supporting cells, and in particular, we're studying how they sense damage and control hearing sensitivity. So today's talk, we're talking about the initial protagonists, the cochlear supporting cells, but in particular, how these supporting cells sense damage via activation of TRPA1 channels. And as mentioned earlier, this work actually started as a Hearing Health Foundation grant, and right now continues through support from the University of Kentucky College of Medicine and Department of Physiology. So what is TRPA1? TRPA1 is an ion channel. It's a master sensor of tissue damage. The channel is formed by 4 subunits of the protein. So here's one of the subunits that make the channel. Each subunit has this long intracellular tail that has lots of cysteines in the tail and lots of anchoring repeats. Those cysteines are the places where a lot of reactive agonists will bind and lead to the gating and opening of the channel. A lot of these reactive agonists are produced downstream of membrane lipid damage that is caused by reactive nitrogen or oxygen species. For example, today we'll use a lot 4-HNE throughout the talk. 4-HNE can directly bind to these cysteines, open the channel, and then it's a non-selective cation channel, so lots of positive channels will go inside the cells. These channels are also activated downstream of other receptors for other substances like prostaglandins or bradykinin that can be produced in inflammatory conditions. So an inflammatory soup where there's a lot of vasodilation. Some of these agonists can directly bind the channel, or they can bind their specific receptors, and downstream of those receptors, downstream of G protein coupled receptors, some signaling can lead to the gating of TRPA1 channels. Lastly, TRPA1 channels also have an EF-hand-like domain, so calcium ions can directly bind and gate these channels. So collectively, the channels can sense lots of different ways damage that's happening in the tissue. And they are highly expressed in nociceptive neurons. So all over the dorsal root ganglion neurons and the trigeminal neurons also right in the face. So when these channels are activated, they code for pain-like signals. They're also expressed in the cochlea, but 2 different groups back to 2006 generated TRPA1 deficient mice, and both groups reported that these mice had impaired chemical, mechanical, and cold nociception, but they had normal hearing, normal balance, and normal mechanical transduction. So we had a channel that we knew was highly expressed in the cochlea, but we didn't really know what it was doing there. So here's a little bit of that review on the expression of these TRPA1 channels. Initially, the laboratory David Corey showed some in situ hybridization against TRPA1 gene. And you can see in blue along a cochlear cross-section, some regions in the cochlear epithelium that are highly expressed in TRPA1. If you zoom in asterisk, here are the hair cells, inner hair cells, and the 3 rows of outer hair cells that were identified through other in situ hybridization against other genes that are highly expressed in the hair cells. And then TRPA1 was here pretty close to the outer hair cells. Some newer data from the Matt Kelly lab using tissue from mouse at postnatal day 1 and single cell RNA-seq shows that the TRPA1 expression is mainly on this type of supporting cells called the Hensen cells, not really in the inner or outer hair cells, maybe a few Dieter cells. So some of the cells around these hair cells, but not the hair cells themselves. And more recently, just a couple of years ago, Dr. Taylor Clark regenerated a new reported mouse because it's been really hard to find protein labeling of TRPV1 in the cochlear epithelium. Pretty much all commercially available antibodies will label the knockout tissue. So we cannot really trust the antibody labelings to identify where TRPV1 is expressed. So in this case, he generated a new reported mouse where the stop codon of the TRPV1 gene was removed and replaced with a flippase sequence. So all cells that express TRPV1 will now express also this recombinase. And then these mice were bred with mice that express TD tomato expression that's sensitive to the flippase activity. So all cells that express TRPV1 will now express TD tomato. And in the cochlear epithelium, again, he found this TD tomato expression close to the hair cells, but none on them. We repeated this experiment using these mice, but now we added labeling for the hair cells because we wanted to confirm where the TRPV1 was expressed within the cochlea. Here are the different turns of the cochlea of adult age mice. In green, you can see the rows of hair cells. In blue, the nuclei of all the cells in the tissue. And in red, the TD tomato signal that's driven by the TRPV1 expression. And again, it looks like only the Hensen cells, so the cells next to the outer hair cells, are expressing TRPV1. And consistent with this data, we previously showed that functional TRPV1 activity is in this region of the cochlea. To do that, we did some live imaging. We took cochlear epithelium from mice. Here in the top is a bright field image. You can see the rows of hair cells, they're very organized. And here within these yellow dashed lines are the Hensen cells. This yellow arrow shows the position where we put a pipette and we deliver locally different agonists for TRPV1. And in the same region, we perform calcium imaging. And this ratiometric calcium imaging allows us to identify changes in intracellular calcium levels at different cell types. So here in blue and purple, it's a low level of calcium, but as the cells increase calcium, you will see green, yellow, or red. I'm going to play a movie, and the arrow will show up here at the position when and during the application of the TRPV1 agonist. This movie is played about 50 times faster than normal speed. So here the arrow shows the delivery of the agonist. The agonist is not applied anymore, but these calcium responses remain for a long time. So repeating again, slow generating calcium responses, but once they're generated, they're very robust and very long lasting. And here are some pictures of the same movie showing the bright field, again, identified the Hensen cell region. The Hensen cells are the ones that show these calcium responses that continued even after we stopped the application of the agonist. And here's just a trace showing for one individual cell, the type of behavior we see. A slow generating response, but very robust and very long lasting. We observed this type of responses with multiple agonists, specific with TRPV1, but in all cases, when we check the tissue for mice that lacked TRPV1 channels, we did not see any of these calcium responses. And like I just mentioned, we evaluated about 5 different TRPV1 agonists. Now, we were applying the agonists in this region, the Hensen cell region. And here's just another example of these calcium responses that we see in the Hensen cells. But sometimes we saw calcium responses on this other part of the cochlear epithelium. This part up here is the Colliker's organ. So we were applying the agonist here in the Hensen cells, and every now and then we saw some responses in this region. The responses were different. They were very fast growing calcium responses, but short-lived. So they decayed really fast. The speed of propagation of these responses was 20 to 30 times faster than the speed of propagation of the Hensen cells. When we moved the pipette to this region and applied the agonist, we did not see any responses. So it was not just diffusion of the agonist to this region. We saw, in fact, propagation, cell-to-cell propagation from the Hensen cells to this region. Here in the top panel, it looks like the calcium wave just jumps. There's no signal coming from here. Here we have the outer hair cells and the inner hair cells, and those cells were loaded with the calcium dye. However, we had a really hard time loading the pillars and diter cells with our calcium dye. So it looks like the signal just jumps. But in the cases where we did have some calcium dye loading the supporting cells, we saw cell-to-cell propagation between these cells. Excuse me. Sorry about that. So we saw propagation between the supporting cells from the Hensen cells. into the colliculus organ. But the calcium waves in the colliculus organ were very different. They were very fast. And I'm not showing today, but we did experiments in the presence of ATP receptor blockers. And we saw that these waves no longer were present. The propagation happened, but stopped here near the inner hair cells. So we know that these TRPA1 dependent calcium responses were propagating and triggering responses that were previously reported by other labs. These are ATP-dependent cell-generating calcium waves. So we have now these ATP responses that, these TRPA1 responses that propagate along different types of supporting cells, always in this direction. We actually never saw propagation in this direction. And we think that's mediated due to the different presence of different types of gap junctions between the different types of supporting cells. So why are these calcium waves propagating? What are they doing? Why is that important? Well, we also looked at the videos we took to bright field imaging that we took simultaneously with the calcium imaging. And again, we have here the rows of outer hair cells, pillar cells, Hensen cells. And I'm going to show a video of bright field, this time 100 times faster than the acquisition speed. And when a white arrow will show up at the moment when the TRPA1 agonist is delivered. So here's the liver. And then you can see the whole tissue looks like it's moving during the application of TRPA1 agonist. Remember that this video is played at 100 times faster. So these are very slow and long-lasting motions that are happening in the organ of Corti. We measure the movement before the delivery of the agonist versus the movement after. A value of 1 will be no change, and that's what we saw in the tissues that lack TRPA1 channels. But in the wild-type tissue, we saw a significant increase of movement after the application of the TRPA1 agonist. So activation of TRPA1 channels in Hensen cells was leading to movement in the tissue, but we still didn't know which cells in particular were leading to this movement because it looked like everything was moving. So we performed multiple images at multiple planes along the tissue and try to quantify cell diameters and movement of the tissue. And eventually, when we imaged really close to the basilar membrane, so at the base of the tissue, we could see the diameter of the pillar cells. In this picture, these are hair cells, but we also got pictures of the data cells down here. And we were able to confirm that diter cells and pillar cells were contracting after the application of TRPA1 agonists. It was not surprising to see that for diter cells, since it was previously reported that these cells have calcium-dependent contractile machinery, but it was quite surprising to see pillar cells contracting. They have been assumed to be very stiff and kind of static, maintaining the tunnel of Corti. So we were not expecting to see anything there, so we wanted to confirm this. And to do that, we decided to patch the pillar cells. And in the intercellular solution in the pipette, we put caged calcium. So calcium trapped inside a molecule that upon UV stimulation will be released. That way we could control release of calcium in individual cells. And we perform calcium imaging simultaneous with bright field imaging to compare calcium levels and cell diameter. So in fact, after UV stimulation, we could release that calcium in just that one cell that we were patching. Here's an example of that increase in intercellular calcium upon the UV stimulation, and the cells when we didn't have caged calcium in the pipette, we did not see an increase in calcium. And again, in the cells where we did have caged calcium, we saw a significant reduction in diameter. So we were able to confirm that pillar cells do have some calcium-dependent contractile machinery. So overall, so far, we knew that Hensen cells are the master sensors of this damage, at least from the endolymph side. They express TRPA1 channels once there's tissue damage, for example, after reactive oxygen species. The TRPA1 channels are gated, and then they activate diters and pillar cells through some propagating calcium waves. Then diters and pillar cells contract, and we saw that reticular laminate actually got elevated. So they contract the tissue and push the bundles up. And others have shown that supporting cells can change shape upon multiple cochlear insults, NOx exposure, damages to stria. So we're not the first ones to show this type of supporting cell changes. But here we wanted to now show how is the change in the organ of Corti and change in the supporting cells, is that really affecting hearing or not? So we propose to use noise exposure because we know noise leads to an accumulation of reactive oxygen damage in the tissue. It's been shown that one round of noise exposure can increase reactive oxygen species for days or weeks after the exposure. We know that this reactive oxygen damage will damage the lipid membranes and lead to the production of byproducts like 4-HNE that can directly bind and engage TRPA1 channels. Then we wonder, in after noise exposure, how is hearing going to change in mice that express or do not express TRPA1 channels? So in mice that contract or do not contract the tissue downstream of activation of TRPA1. And in fact, we confirmed this previous results showing that one round of noise exposure leads to an accumulation of 4-HNE in the tissue. This is an immunolabeling of 4-HNE modified proteins. 5 days after noise exposure is where we see kind of the peak after our type of noise exposure. 2 weeks later, it's clear, but not quite reached baseline levels. So it's a very long-lasting damage in the cochlear epithelium. Here's the quantification of that increase at 5 days. So first we check the hearing. This is just the wild-type mice in black. We're looking at hearing thresholds along different frequencies. Lower numbers mean better hearing. So mice are really sensitive in the 16 to 24 kilohertz region, and less sensitive below or above that. The TRPA1 deficient mice, as reported previously, had normal hearing thresholds. So they hear the same, they hear normal. Now, after noise exposure, we use 30 minutes, 100 dB SPL, broadband noise. Here I'm plotting the threshold shift. So this is the changes from baseline. So at 0 will mean that after noise exposure, there were no changes. Here for 8 kilohertz, there was about a 10 dB increase. For 16 to 32, we're talking 40 to 60 dB increase in hearing thresholds. This is immediately after noise exposure in the wild-type mice. And this in red is immediately after noise exposure in the TRPA1 deficient mice. And we do see a significantly larger increase in some of the frequencies. However, 5 days later, hearing thresholds start recovering, but they have recovered better in the TRPA1 deficient mice. So if we look at a particular frequency over time, it looks like there's no difference before noise exposure. Immediately after noise exposure, the mice that lack TRPA1 channels lose more hearing. 5 days later, those mice recover better. I'm not showing here, but 2 weeks later, we have same hearing thresholds. 8 and 16 dBs had full recovery, while 24 and 32 no dBs kilohertz had some permanent threshold shifts. But overall, we had this faster recovery that we were able to quantify by looking at the difference between immediately after noise and 5 days after noise. And quantifying this, we see in most all frequencies that we tested, except 8 kilohertz, the TRPA1 deficient mice had a faster recovery. And consistent with this data, we also evaluated distortion product optoacoustic emissions. Here's just a measurement of cochlear amplification. Before noise exposure in the solid lines, we see no difference between the wild type and TRPA1 deficient mice. Here in the x-axis is the sound intensity. So at more intensity, you have more emissions. And this is for the 16 kilohertz region, so that region where they're most sensitive. 5 days after noise exposure in the dashed lines, the TRPA1 wild type mice had barely no DPOAs yet. So the cochlear amplification is still down. They still have elevated hearing thresholds, while the TRPA1 deficient mice have started recovering. So they're recovering faster. This is consistent with those better hearing thresholds that I just showed. So overall, right now, we see that before noise exposure, there's no difference in hearing between the mice. Immediately after, we have a difference, and I'll go into a second about how we think this is happening. But 5 days later, we see a faster recovery in the mice that lack TRPA1 channels. We think that this increase in hearing thresholds in the wild-type mice is due to the activity of TRPA1 channels downstream of those 4-HNE, all that reactive oxygen damage that's being generated in the tissue, which peaks around 5 days in our model, and it takes over 2 weeks to clear. So TRPA1 activity is the highest within this like 3 to 5, 3 to 7 range window. And at that point, we think TRPA1 activity through the contraction of the organ of corti of the supporting cells is keeping hearing thresholds elevated, maybe to allow the tissue to heal while there's still some reactive oxygen damage happening by kind of inhibiting some of that sound perception. Now, immediately after noise exposure, we do see what looks like a protection also by TRPV1 signaling, because in the absence of TRPV1 signaling, there's more hearing loss. We think at this point, there's no 4-HNE being generated. So how is TRPV1 being activated? We think it is downstream of ATP, because ATP has been shown to be released into cochlear fluids during sound exposure, during noise exposure. So loud sound will lead to this release of ATP into the fluids. And I'm showing here now, we showed that ATP can activate also TRPV1 channels. So in this experiment, we use HEC293 cells transfected with TRPV1 channels in GFP, so we could identify the cells that were expressing the TRPV1. And here, AITC, or mustard oil, is an agonist of TRPV1 channels. When applied, leads to an inward current, so gating of the channels in these cells. The cells that were not transfected or mock transfected just with GFP did not have these currents. And here's just the IV plot, the current voltage relationship, showing that only the cells that had the TRPV1 construct responded to the TRPV1 agonist. Now, in the same cells, now, if we deliver ATP instead of the TRPV1 agonist, ATP was able to trigger that same gating of these TRPV1 channels. And ATP led to small responses to ATP receptors that are endogenously expressed in the cells, but very tiny, unlike this TRPV1 currents that we were able to see. So I'll just go back here. So again, we think that ATP that's released in cochlear fluids during the noise exposure, this is a very fast way to activate TRPV1 channels to start protection during noise exposure. But later on, ATP is clear, and this reactive oxygen damage is the one that continues activating TRPV1 in the tissue. So lastly, we looked at whether TRPV1 was in fact protective or not. Like I mentioned, 2 weeks after noise exposure, we did not see big differences in hearing thresholds between the two groups at the noise exposure levels that we used. What we did see was some changes in the latency of this auditory nerve responses. So we look at the wave 1 of the auditory brainstem responses of the mouse. This first large peak is the auditory nerve activity. And if we look at the time that it takes for sound to activate the auditory nerve, we call that the latency of the wave 1. In the solid line is the mice and wild-type mice before noise exposure. In dotted line, we have 2 weeks after noise exposure. This noise protocol is a broadband noise that leads to full recovery at some frequencies and permanent threshold shifts and other frequencies. And this is the responses to a click stimulation, so a mixture of frequencies. And we see a permanent increase in this latency. And you can see here in the x-axis, this is sound intensity. As the sound is louder, the latency gets smaller. So an increase in latency typically correlates with some hearing loss. But here, this click ABR wouldn't tell us exactly which frequencies. What's interesting is that in the TRPV1 deficient mice, we saw that similar increase, although a little bit larger, at these low intensity sounds. But at high intensity sounds, we see now a permanent decrease in latency. So a permanent decrease in latency means that the traveling wave in the cochlea is either propagating faster and reaching the places in the cochlea faster, maybe to an increase in stiffness in the tissue, or the tuning curves in the cochlea have changed, and now frequencies are detected at different positions in the cochlear partition. So we decided to study that a little bit more, and now instead of applying click stimulation, we applied now tone-specific stimulation, 8, 16, 24 kilohertz, and we also tried different noise protocols. And in this case, after 30 minutes of 85 dBSPL broadband noise exposure, we had full recovery at all frequencies in hearing thresholds. So this is just a temporary threshold shift protocol. And we're looking at the latency of wave 1, and we had full recovery 2 weeks after noise exposure in the wild-type mice. So in solid line is before noise, and dotted line is 2 weeks after. Everything recovered fully 2 weeks later. In the TRPV1 deficient mice, however, we do see a significant decrease in the latency of the wave 1. Like I mentioned earlier, if you are looking at hearing loss, this latency gets bigger as the sound intensity decreases. So hearing loss should lead to an increase in latency. But a decrease in latency, a significant decrease in latency, it's either due, we think, to changes in the stiffness of the tissue, or changes in the tuning curve. And this is where my lab is currently exploring, trying to understand what is leading to these changes in the stiffness in the cochlear partition of these TRPV1 deficient mice. So in summary, today we talk about multiple types of supporting cells, multiple types of cells in the cochlear epithelium. We have that Hensen cells are the master sensors of tissue damage in the cochlear epithelium, at least the damage sensed through the endolymph side. They express TRPV1 channels. They have long-lasting calcium responses that are slowly propagate through Dieter cells and pilar cells. Pillars and Dieter cells have calcium-dependent contractile machinery. So when these calcium waves reach them, they will contract and actually lead to a raise in the reticular lamina, and the whole organ will get smaller and change how sound detection occurs in the tissue. So initially, mice have normal hearing where they have or not TRPV1 channels. Immediately after noise exposure, there's a protective signaling downstream of TRPV1 channels. So the mice that express TRPV1 channels don't lose as much hearing. But then the mice that don't have TRPV1 channels recover faster because there's lack this temporary threshold shift type of signaling that TRPV1 channels are mediating through the contraction of the tissue. 2 weeks later, even in the mice where we had full recovery of hearing thresholds and no difference between wild type and knockouts, we did see changes in the latency of the wave one of the ABR. So the auditory nerve activity was delayed. It was delayed in the wild type mice, and it was occurring now faster in the knockout mice. So we think there's some changes in hearing quality also in these mice. Now I want to finish just by thanking my lab members. Here are pictures of the lab over the years, the current members of the lab, all my collaborators. I'm just listing here on my social media in case anybody wants to connect, and happy to take questions now.
Well, what a beautiful body of work, Dr. Vélez-Ortega. I mean, those pictures of active movement of calcium that you showed and how it goes from Hensen cells all the way to the organ on the other side. And what you've shown is that there's a relationship between this particular receptor as well as hearing loss or damage. And by the way, I want to remind our audience that they can submit their questions in the Q&A box at the bottom of their Zoom. Do you think, how is the hearing loss occurring? Is it damage to the hair cells? Is it damage to the hair cells, so there's calcium flux that's going on? Or what's the relationship between the supporting cells and how they're responding to noise and what we see in terms of whether it's otoacoustic emissions, whatever is ABR? How are the hair cells in turn related to the TRPA1 mechanism?
Yeah, we're still working on it. And first we're looking at what type of damage is happening. When so we have these changes in the latency of the ABR, and we're trying to check whether it's a problem with the turning curve, or something happening more in the mechanics or the stiffness of the tissue. But we have looked at ribbon synapses in the hair cells to see if there's some type of hidden hearing loss, since we see full recovery of hearing thresholds after one round of noise. And we haven't seen anything big in the inner hair cells, but we have seen now some differences in the outer hair cells. We're seeing differences in ribbon volumes and ribbon placement. So it looks like there is some small damage happening in the outer hair cell side, but we don't know exactly how those changes in ribbons will relate to permanent hearing loss yet. But we haven't looked at other types of damage yet. So we still haven't looked carefully, maybe our stereocilia, and see if anything's happening there. We do have some previous results from the lab where we do see some ATP-dependent currents in hair cells. And initially, we thought the TRPV1 channels were expressed in the hair cells, but the more we look at it, it doesn't look like they express TRPV1 channels. So perhaps while these calcium waves are traveling and the supporting cells, maybe somehow they can activate the hair cells as well. But we haven't found that how or if that's really happening yet.
Well, that was going to be my next question. Is there a secondary impact of the calcium as it, traverses from the supporting cells? We know calcium is involved in signaling, of course, in the hair cells and so on. So um, there's a question: do mice less than 6 weeks old show a different response? Is there a different response to the different ages in these mice?
Yeah, a lot of our calcium imaging was done in neonate tissue. We did do some around postnatal day 20 to show that in more mature cochlear tissue, we still have Hensen cells having these big, long-lasting calcium responses, and they're still there. But most of the studies in calcium responses and how they propagate was done neonate because it's really hard to study that in the adult tissue. We have looked now at hearing function differences, whether mice around 3 to 4 weeks of age, 40 days old, have different responses versus mice that are more of 4 to 5 months old. We haven't found anything big different between them yet, but we haven't evaluated whether noise exposure would maybe trigger some age-related hearing loss in those TRPA1 deficient or not, or wild-type mice. That's an experiment we haven't done yet.
Do you think that your TRPA1 channel and the mechanisms associated with noise exposure may in part explain cochlear synaptopathy? Is there any relationship between what's being, examined by the Harvard group and what you're studying? Is there a relationship there?
Yeah, we looked at the inner hair cells and we did not see any signs of loss of ribbon counts yet. But we might have data supporting changes in the volume of the ribbons right now. We're trying to confirm that. So maybe there's a little bit of synaptopathy, or we checked too early, I guess. Maybe not. We didn't wait maybe long enough to see if it's fully developed, but there might be something there. Yeah.
Gotcha. And we knew hair cells danced, increasing and decreasing in length. We didn't know supporting cells did until you just showed those wonderful pictures. What exactly is the implication of these cells also changing in size? And yeah,
Yeah, I'm fascinated by the morphology of the tissue, how the whole tissue is built. So Dieter cells are underneath the hair cells, and they have a phalangeal process, a little arm that grabs hair cells on top, but doesn't even grab the same hair cell. It grabs like 3 cells down. And this phalangeal process is the one that's been shown that can easily move with changes in calcium. Now we're showing that the cell body is also contracting. So that will change the geometry. That will change the angle of the hair cells and potentially will change how much the hair cells can dance because it might put some constraints and inhibit some of that amplification that way. We were very surprised to see that the pillar cells also contract. We were not expecting to see much change in those cells. Um, and that will change. We see an raise in the reticular lamina. So that is easily explained by just contracting these pillar cells will just push the whole tissue up. And consistent with also reports from other groups showing that contraction of the organ of Corti. We think they're just constraining the vibrations and the organ of Corti by doing all those contractions. So dampening our hearing sensitivity. And I think it's probably to allow the tissue to recover. There's a lot of tissue damage happening. So less vibration is probably better if you want to heal some of that damage.
well, this whole thing about tonotopicity in the cochlea. Do you think this contraction of the supporting cells also plays a role in addition to all the other things? The stiffness of the basal membrane, the outer hair cells, and so on.
Mhm.
Or do you think that's not really related?
I think if we change the stiffness of the tissue at any position, we will change definitely the tonotopic arrangement of the cochlea. And I think that will be reflected in changes in turning curves at different positions. Mhm.
And I think that'll be very interesting to look at that. And there's a wonderful question about glutamate. We know that with excess accumulation of glutamate, there can that activate your TRPA1 channels by any chance? Because you see that
Oh,
with noise exposure as well. And have you looked at that?
yeah, where glutamate can activate TRPV1 channels. I don't know if that could happen. I don't know if there's any evidence for it. We didn't test it, but in noise exposure, you definitely have all that glutamate like toxicity that could happen. So I don't know. That would be good to check.
It's a great question. It's from an
Mhm.
anonymous attendee, by the way, in case you didn't know who that was. It's a good question.
Mhm.
There is an interesting question from one of our staff members. They're wondering, you mentioned in the beginning that these TRPV1 channels are involved in pain perception and so on. So this is a stretch, a big leap now. Is
Mhm.
hyperacusis or other inner ear pain syndromes related to this channel? And is there a way to block this channel to test that hypothesis?
Yes, that's I'm very interested in that question. And this is wild speculation, but if TRPV1 channels are these pain sensors, and I'm claiming here lack of TRPV1 channel will make you more susceptible to noise-induced hearing loss or noise-induced hearing damage, then people that have maybe an abnormal TRPV1 signaling um, might be more susceptible to noise-induced damage or noise-induced hearing loss. And pain sensitivity or pain perception is so variable between individuals. So people that maybe have higher pain thresholds, maybe because of defective TRPV1 signaling, might be living a very happy life because they don't feel pain as much, but they might be more susceptible to this noise-induced hearing loss. The only two mutations that have been identified in TRPV1 are both gain-of-function mutations. No surprise because they lead to a phenotype of pain syndromes. But there haven't been any lack of function mutations in TRPV1 identified, but they're definitely being reported. Mutations that should lead to a loss of function, like early stop codons, things like that. So I think there are definitely undiagnosed TRPV1 defects and could potentially lead to severe sensitivity to noise-induced hearing loss or some potential age-related hearing loss.
Well, in these times of human relevance of all our experiments, that's certainly a good one right there. You know,
Yes, yeah.
how do you know that the that the flux in calcium in the inner and the outer hair cell synapses are not triggered in the calcium release experiments? That is, that there could be other sources of calcium, or are the hair cells not releasing their calcium in your experiments?
In our experiments, we have calcium dye loading the hair cells. And when we applied the TRPV1 agonist in the endolymph with the intact tissue, we don't see responses in the hair cells. However, when we disrupt the tissue, so we allow this TRPV1 agonist to get on the basolateral side of the cells, we start seeing some hair cell responses. But again, we now don't see expression of TRPV1 in hair cells. So we wonder how those responses could be happening, and is it downstream of something in the supporting cells that's being released and then activated somehow the hair cells. So we're still very early trying to explore those connections between the supporting cells and the hair cells.
Gotcha. And and a follow-up question from our anonymous attendee who states that they have hearing loss. Um, is there future therapies that will be, mediated through modulation of this particular channel for, let's say, noise-induced hearing loss or age-related hearing loss or any of those things?
Yeah, so ideally, right, you will take something before you go to a nightclub when you're going to be exposed to noise exposure, something that will prevent that damage. Activating TRPV1 mediated signaling, it's probably not the way because then it's a pain sensor, so it will be activated. Pain signals all over your body. So what we need to find is downstream of TRPV1 signaling, where in that pathway of supporting cell contractions, where can we activate that pathway without having to activate TRPV1 channels? And that's why I'm really interested in dissecting all of those steps. After TRPV1 signaling, what in the Henson cells, it's what's activated that will then go from cell to cell, and where in that pathway we can find a pharmacological way to trigger it, hopefully before noise exposure to prevent some damage in the tissue.
Gotcha. Now, I think you have answered this question already, but I think I'm going to ask it in slightly a different way because two different attendees are asking this question.
Mhm.
What is the potential cause of increased latency of wave one And did you get a chance to look at the IHC synapses? And I think you said you did, and there were no changes, but please go ahead and there's interest in
Yeah.
that question, so you might as well attack it again.
Yeah, an increase in latency will correlate with some hearing loss. So increasing hearing thresholds will correlate with an increase in latency. What we see in the triple one deficient mice is a decrease in latency. And that's what really, we repeated several experiments, several cohorts, and indeed we keep seeing an decrease in latency. And a decrease in latency typically means so the traveling wave enters the cochlea at the base and takes some time to reach each position. We see that significant decrease in latency. We move away from the base of the cochlea. So when we go to the lower frequencies, we see it clearly. So either the traveling wave is moving faster in the cochlea, which could happen if the tissue is stiffer, or the tuning curves, instead of being sharp, are now wider. So sound reaches and activates hair cells sooner than before. So those are the two hypotheses that we're exploring. We have not seen differences in the counts of inner hair cell synapses. So it doesn't look like, initially it doesn't look like big synaptopathy, but when we do look at the volume of the synapses, we do see some changes. They're getting smaller. So we're repeating some of those experiments and looking more carefully at the synapses because something might be happening there, but that wouldn't explain the decrease in latency that
Yeah.
we see.
Well, there's a lot of congratulatory notes on the greatness of your work, so I want to make sure I communicated that from our attendees.
Thank you.
Uh, they wonder what's the strain of the mouse that you're using in your experiments and if you looked at different strains.
Yes, most of this work is in C57 Black 6. So that's why our older cohorts were no more than 4 or 5 months because we have this hearing loss in that strain. We did do some experiments initially on a different background. We have done a couple on different backgrounds and everything behaves the same. But if we want to move towards noise exposure and some more aging experiments, we will need to move this mutation into a different strain for sure. But most of the data showed today is from C57 Black 6, although a lot of the key results were confirmed in other strains.
So I'm surprised that nobody's asked this question, and this may be just a stupid question, so I'm going to go ahead and ask it. So the hair cell regeneration is thought to occur from supporting cells, and now you're showing that there's some role of the supporting cells in response to noise trauma or how they respond to it. And any connecting the dots there at all in terms of maybe this is the reason why these cells are detecting the damage as well and then responding potentially down the road in other species with hair cell regeneration, or do you see a connection or not?
That's a good question. I don't even know if triple one is expressed in other sensory epithelium from species that regenerate hair cells. So I will have to start there. I mean, we were surprised that TRPM1 was not expressed. So far, it doesn't look like it's expressed in the paraganglion neurons. So it's a receptor that's typically expressed in nociceptive neurons. We thought it was just going to be in the sparaganglion type 2 neurons, and apparently it's not there. So we were quite surprised to see it in the supporting cells, but only in the Hensen cells, apparently not everywhere. So I wonder if there's other species that have also TRPM1 expression in supporting cells. They might not even express it. So I don't know. I'll have to double check.
And there's no neural markers in Henson cells, right? Besides this particular synapse?
We are looking, and some things not really highly expressed, but nothing big comes to mind right now. No, but we're trying to look what else are they expressing that will help us understand how they are connecting to other cells and communicating signals, and whether they can even communicate through type 2 sparaganglion neurons.
Gotcha. And so since you talk really focused on supporting cells, and there's lots of other types of cells within the cochlea, any connection to the stria vascularis or any of the spiral ligament or any other beautiful, or even the lining of your, the endolymphatic chamber or something. Anything else that you want to share with us in terms of the, you know, non-hair cells that might be also of interest to you?
Yeah, I'm in this tria. There's There's some literature with TRPV1 expression in endothelial cells, but a lot of that is with antibodies that we don't truly trust. But in these mice, with the reported, I don't see TRPV1 expressed in the stria either. So it seems to be very localized to hence the cells, but there's definitely reports of damage coming from the stria leading to
Yeah.
contractions of the organ of Corti through supporting cells. And that maybe could be mediated through TRPV1 being activated. Damage the stria should lead to also reactive oxygen damage, and it could activate TRPV1 too.
Yeah, I just want to make sure our audience didn't forget the the stria as
Yeah.
well. Just the other non-hair cells within the cochlea. There's
Mhm.
a question, is there evidence in your data for, quote, toughening of the cochlea after noise exposure?
I'm thinking that toughening is more resistance, I guess, against further noise exposure. We tried that. We actually tried, okay, what if one round of noise exposure, if you have triple one channel, protects you against additional noise exposure? Um, and we did find some data supporting that, um, but it gets confusing depending on how soon you expose the mice, right? During the peak, when triple one activity is at its highest, yes, definitely a second round of noise exposure, they're more protected. But at that moment, there's also slight changes in hearing thresholds. And then trying to now normalize to that, delivering different noise intensities to the different groups of mice became a little too complicated. So we're still working, I guess, on that question, but it looks like yes, it does protect when triple one activity is active.
Well, I think we're coming to the end of our hour here. Your wonderful talk put the spotlight on some of the other cells within the cochlea, which is really
Thank you.
great. It's good that the lead singer isn't the only one that gets an attention in the band. The supporting players and stuff, and
Mhm.
the chorus are getting one. So we thank you so much for your wonderful talk. Um, we, of course, are so grateful to you, our community, for all your support of our Emerging Research Grants program. And please remember that you can donate to our efforts to advance better treatments and cures for hearing imbalance conditions at hhf.org/donate. And thank you, Dr. Vélez-Ortega. Thank you, our audience. Thank you for all those questions. Please enjoy the rest of your day.
Thank you.